you for your
Thank you, Dr. Radovich. For those that are interested in receiving CME credit from this presentation, please go to the URL link shown on this slide if you're part of the BioConference Live. Next, I'd like to just remind everyone who is attending this presentation that the products shown by Dr. Radovich from Life Technologies are for research use only and not for diagnostic procedures. Also like to recommend that if anyone is interested in learning more about RNA sequencing, transcriptome, the ion torrent proton, there's information available in the Life Technologies booth. At this point, as there does not appear to be any additional questions, I'd like to thank Dr. Radovich again and everyone who attended the presentation. Thank you. Thank you for joining. My name is Michaeline Bunting, and I will be the moderator for today's presentation. On behalf of Life Technologies, it's my sincere pleasure to introduce Dr. Milan Radovich, Assistant Professor of the Department of Surgery, Indiana University School of Medicine, a member of the Melvin and Bren Simon Cancer Center and Center for Computational Biology and Bioinformatics. Dr. Radovich will be speaking on the transcriptome sequencing in translational oncology research. This presentation is available for CME credit, and I will have more information available at the end of the presentation. We'll also be taking questions and answers during the presentation, and I'll be facilitating that session at the end. Dr. Radovich.
Okay, Michaeline, thank you so much for the fantastic introduction, and I'd also like to thank Life Technologies and the organizers of BioConference Live for allowing me to present this webinar today on transcriptome sequencing and translational oncology research. As a quick overview, I'm going to give a very brief introduction to cancer genomic sequencing and why we perform next-gen sequencing in oncology research, and then delve into our primary focus is work in genomic sequencing in triple-negative breast cancer, and then end off with some of our work in applying it as correlatives in clinical research studies. So when I give a talk to a varied audience, I always like to start off with this slide to kind of go over the history of how genomic sequencing has evolved over the decades. Many on this webinar will probably remember the days of sequencing DNA using gel-based terminator sequencing. And as the technology evolved into the 2000s, we moved into capillary-based sequencing. As many would remember, these sequencers were the workhorses of the Human Genome Project, churning out millions of bases per day at 3 major genome centers. After the advent of, after we got through the 2000s and we came upon the advent of next-generation sequencing in 2005, we saw just a plethora of instruments come out onto the market. Coming now to today, these instruments producing on orders of billions of bases per day, and in the near future, the advent of nanopore technology and single molecule technology. Why we're so interested in the idea of using genomics in studying cancer is that for as many people know in this room, the purpose of, or the cause of cancer is a DNA-based disease. We know that cancer in its essence is due to mutations to the genomes of cancer cells. So this figure published in Nature a couple years back is a fantastic figure describing this accumulation of cancer mutations as they're caused through a variety of processes. First, there are intrinsic mutation processes, so as cells move from fertilized egg to gestation, as infancy, childhood, adulthood, and clonal expansion, we know that there are intrinsic mutations that are generated. These are mutations that are generated due to just the DNA replication machinery. Though that machinery is quite good at what it does, it's not perfect, and every once in a while mutations are introduced. We also know that there are mutations that are introduced due to environmental and lifestyle exposures. Things such as smoking, sunlight, and a variety of carcinogens also add mutations to the genomes. But we know that many of these mutations are actually what we call passenger mutations, as depicted by these yellow and blue dots. Mutations are introduced in the genome but cause no change in protein function. But what unfortunately happens is the accumulation of driver mutations. These are the ones present in stars. These are mutations that affect tumor suppressors and oncogenes, and as they accumulate, begin to transform the cell from normal cell to early clonal expansion to benign tumor, early invasive cancer, late invasive cancer, and then obviously what we're most concerned with is chemo-resistant disease. We also know, with recent data published in major journals like New England Journal of Medicine here, is that tumors themselves are highly heterogeneous. In this fantastic paper published in the New England in 2012, a sequencing of a large renal cell tumor where the investigators took multiple punch biopsies at 10 different sites within the tumor, and also within the chest wall metastases and the lung metastases, and then sequenced each of these sites. And what came out of this work was a very interesting observation that within these various sites, there are mutations that are shared among all the parts of the tumors and the metastases, but there's also mutations that are shared only in the primary that are not seen in the metastases and vice versa, but also mutations that are specific to each physical site within the tumor, engendering that a massive amount of heterogeneity mutationally is present in these tumors, such that they actually form an evolutionary tree. And as these tumors evolved and as they continue to grow into these large masses, actually a microcosm of evolutionary divergent evolution occurring in which mutations are selected for and gained advantage as the tumor grows and then begins to metastasize. We also know that these mutations, these accumulation mutations in the key tumor suppressors and oncogenes then translate into differences at the RNA expression level. Here I show a principal components analysis of some example data of 10 normal tissues and 10 tumor tissues, where we see, based on RNA-seq expression, the principal components analysis, for those who are not familiar, is basically a 3D representation of gene expression on a global level, such that samples that are more alike on their gene expression level are balled together, and then samples that are more different or disalike based on their gene expression level are separated. And here we can see a wide separation between the tumor and normal samples. And it's not uncommon to see a dysregulation and expression of thousands of protein-coding genes and a transformation of normal cells to tumors. With the advent of all of this sequencing technology, both DNA and RNA, the National Cancer Institute has instituted the Cancer Genome Atlas. As mentioned, this is a large National Cancer Institute initiative to characterize more than 10,000 tumors and 24 tumor types by 2015. And in this initiative, they are applying a variety of sequencing chemistry modalities, including DNA sequencing, RNA sequencing, microRNA-seq, SNP arrays, reverse phase protein arrays, methylation assays from tumor normal pairs, with the goal of creating a comprehensively public catalog of all significantly recurrent genomic alterations. What is fantastic about the TCGA is that its comprehensiveness. Here is actually a truncated list of the major tumor types being considered or being sequenced by the TCGA. In addition to this list, there's a variety of rare tumors that are being sequenced also as part of a rare tumor initiative. Further, what is advantageous about the TCGA is that all this data is made publicly available for investigators to use in the community, both via available the TCGA data portal and also available through the raw data through the UCSC CG Hub. Here, just an amazing resource of next-gen mentioned DNA, RNA, and methyl-seq and variety of chemistries to be used for translational oncology research. So, I've kind of given you an intro to genomic sequencing the cancer, but the question is, why do we do it? I think it's advantageous that we can take these technologies and be able to interrogate at the molecular level in an amazing different amount of facets, but why do we want to do this when we take this to the human level? We know that human cancer is the combination of DNA mutations and RNA dysregulation, and that this combination of mutations and RNA dysregulation is lethal. Also, sobering, the treatment for cancer in the last 30 years has remained largely unchanged. Drugs that are still mainstay first-line therapy for common chemotherapeutics like paclitaxel, cisplatin, and cyclophosphamide were actually discovered in the 1950s and 60s, but still remain mainstay chemotherapy for many patients. And while drugs that specifically target cancer cells are under development, targeted therapies that drastically extend survival have been few and far between. That's not to say that there haven't been a good group of blockbusters such as Herceptin, the aromatase inhibitors, crizotinib, and so on. But those drugs have been very specific to very specific populations, and ones that targeted therapies that impact larger cancer populations have yet to be discovered. So what are some major questions in cancer research? 1, what mutated genes and/or pathways are critical for the development of common cancers? What are the key drug targets for the development of novel therapeutics? What molecular changes cause chemoresistance? And are there better biomarkers for the early detection of cancer? Also, are there better biomarkers for predicting prognosis and treatment outcome? And one of the questions we can ask is, can cancer transcriptome sequencing help answer these questions? So what I'd like to do now is actually show you how we at our group have applied cancer transcriptome sequencing to study triple-negative breast cancer. For some in the audience may already know what triple-negative breast cancer is, but for those who don't, I'd like to introduce the disease and how we define it. So first, a patient with a diagnosis of breast cancer, there's going to be a biopsy of that primary tumor, and that primary tumor biopsy is going to be sent for pathology to have a variety of immunohistochemical stains. One of the first stains that tumor is going to be stained for is for the estrogen receptor and progesterone receptor. About 75% of breast cancers are going to overexpress these two proteins. And in addition to standard chemotherapy, are going to receive drugs that target the estrogen receptor, including tamoxifen, fulvestrant, and aromatase inhibitors. Another 15% of breast cancers are going to overexpress or overamplify the HER2 oncogene, a cell surface receptor tyrosine kinase. And in addition to chemotherapy, these patients will receive drugs that target HER2, like Herceptin, lapatinib, and the newer molecule TDM1. But another 15 to 20% or so of breast cancers are what we call triple negative. These are patients who are negative for ER, PR, and HER2. In addition to standard chemotherapy, there are no targeted agents currently available. This lack of target agents has actually translated into a poor overall survival. Here, a publication by Dent et al. And CCR in 2007 shows an overall survival Kaplan-Meier curve of all other breast cancers versus triple-negative breast cancers. As you can see, patients with triple-negative breast cancers, a diagnosis entails an overall poor prognosis. So what are some details about this cancer? As I mentioned, about 10 to 15% of all breast cancers with a disproportionately higher mortality. It has a higher proportion affecting premenopausal women and women of African descent. We also know that the majority of BRCA1 breast cancers are triple negative, meaning if a patient is a germline carrier of the BRCA1 gene or mutation in the BRCA1 gene, if they develop breast cancer, they tend to develop triple negative breast cancer in addition to being high risk for ovarian cancer. And as I mentioned before, there's a paucity of effective targeted therapies. And the question we ask ourselves is, as scientists, where can we look to better define this disease and define new therapeutic targets? To do this, we embarked on a comprehensive RNA sequencing study where we took 20 microdissected normal breast tissues from healthy volunteers from our Susan G. Komen for the Cure tissue bank at the IU Simon Cancer Center. In addition, we sequenced 10 triple-negative breast cancer tissue samples from our institution. We then merged the data with 84 triple-negative breast cancers and 10 adjacent to tumor normal tissues from the Cancer Genome Atlas in a combined analysis. We merged, normalized, and analyzed the data using a variety of software both developed at our Center for Computational Biology and Bioinformatics, along with third-party software from a variety of vendors, including Life Technologies, Partech, and Ingenuity Systems. One of the most important parts of our study was our use of normal control. Here is actually a picture of an H&E stain of our normal breast tissues, where you can see the blue circular ducts represent the milk ducts present in the normal breast. The importance of this study was that we microdissected our normal ducts and you extract the RNA from the normal tissue, from the normal duct tissue, as our comparator to cancer. The reason being is that the origin of breast cancer is within the ductal epithelium. As also known, the majority cell type that makes up the normal breast consists of fibrous tissue, fat, stroma, and we wanted to enrich for the RNA specific to this area that's the origin of breast cancer. What was neat was these are actual samples donated from healthy volunteers that are present as part of the Susan G. Komen tissue banks and were chosen as part of a larger bank of 1,500 breast normal tissues with matched serum, plasma, DNA, and a full medical history. And we were able actually to capture all that data in our analyses. One of our first results was a principal components analysis of the RNA-seq expression of these various groups. Here in blue are the Komen tissue bank microdissected normal tissues. In green are the adjacent normal tissues from TCGA. The purple are the triple-negative breast cancers from TCGA. And the red represent the triple-negative breast cancers that we sequenced from Indiana University. As you can see in this principal components analysis is a vast difference in the transcriptomic profile between these various groups. And without having the time to go into all the details, we have a considerable number of genes that separate these groups, including roughly 3,000 genes that separate microdissected normal tissue from triple-negative breast cancer. Here is actually the Venn diagram of the number of genes that differentiate these groups. So here in the Venn on the top in the pink, you have the number of genes that separate adjacent normal from microdissected, about 946 genes. The number of genes that separate triple negative from microdissected normal of 3,261 genes. And on the bottom right, the number of genes of triple negative versus adjacent normal tissues at 3,353 genes. One of the analyses that we want to take when we developed this data was to get a global idea of what really differentiate these tissue types. So we performed the canonical pathway analysis comparing these groups. In the triple neg versus microdissected normal, we see pathways including role of BRCA1 and DNA damage response. Like I mentioned, DNA damage in BRCA1 is very important in the etiology of triple negative breast cancer, and including other cancer-specific pathways like cell cycle and cell cycle control over chromosomal replication. But also, something that we also expected was immune pathways like dendritic cell maturation and IL-8 signaling, as it's well known in triple negative that immune infiltration is a major component of this cancer. On the right side, when we compare to adjacent normal, we actually see some of the same pathways we see when we compare to microdissected normal, like BRCA1, cell cycle. But we also see other pathways more indicative of stroma, pathways like atherosclerosis, hepatic fibrosis, and estrogen-mediated S-phase entry. The stark difference in these two controls becomes more evident when you look at pathways that are very specific, though, to both the analyses. So here, these are pathways that of the genes that do not overlap on the Venn diagram. So these include, on the left side with triple neg versus microdissected normal, many immune pathways like antigen presentation, dendritic cell maturation, NFAT1 regulation of immune response. But when we look at the right side, when we use the adjacent normal comparator, we actually get pathways with smaller p-values and ones with pathways not as intuitive, like melatonin degradation, LXR activation. And when in further analysis, when we dug deeper into these, what these were genes involved in activated stroma. Some of the reasoning behind why we may see these differences include the fact that in our microdissected normal, we had actually went through the process of laboriously microdissecting these tissues, where the adjacent normal tissue represents a whole tissue conglomerate. But also the fact that tumors are very well known to secrete a considerable amount of factors into the local microenvironment, such that these factors can play a strong role in influencing the tumor microenvironment and the adjacent normal microenvironment, such as dysregulating genes. And again, when we looked deeper into the data, we saw many cancer genes in the adjacent normal actually upregulated, potentially due to this microenvironment effect. Another analysis that we did was actually perform what we call an upstream regulator analysis. This is a very cool analysis that takes the differential gene expression and then, based on a large literature database, is able to predict whether an upstream activator is inhibited or activated. In this analysis, when we took our differential expressed genes between triple negative and microdissected normal, we present here our top 3 inhibited transcription factors and those that are activated. Interestingly, this data was congruent with known DNA mutational events present in triple negative breast cancer. For example, the transcription factor p53 was inhibited in our analysis. p53 is the #1 most mutated gene in triple negative breast cancer with a prevalence above 80%. We also found CDKN2A or P16 to be inhibited, as common in many cancers, and also RB1 to be inhibited, which has been reported to have a 72% loss of heterozygosity rate in triple-negative breast cancer. Conversely, on the activation state, with RB1 being inhibited, we find its canonical target E2F1 activated. We also find the MYC oncogene activated, and MYC is known to be amplified in a large proportion of triple negatives. And we also, in congruence with the data from the TCGA, find the transcription factor FOXM1 to be activated. This is a transcription factor known to be involved in metastases, and I'll actually come back near the end of my talk to talk a little bit more about FOXM1. So this idea between triple neg versus normal at the molecular level is a very interesting story, but what happens when we take it to the therapeutic level? And the reason we take it to the therapeutic level is maybe to begin to get an idea of some responses of targeted therapies in triple-negative breast cancer. I did mention to you earlier in the talk there are no currently FDA-approved targeted therapies for triple-negative breast cancer. That doesn't mean that it hasn't been tried, and there's roughly now 6 to 7 phase 2 clinical trials testing targeted agents in triple-negative breast cancer. What I show here is actually the conglomerate of those trials, where on the left I show the treatment that was used in the phase 2 clinical trial, the target, the rationale for testing that target based on previous tumor versus tumor data, and then also what we see in the tumor versus normal data. What I mean by tumor versus tumor data is, in many studies, finding targets in triple-negative breast cancer were based by comparing triple-negative breast cancer to other subtypes of breast cancer or to benign disease, possibly leading to potentially identifying a therapeutic target, but also potentially finding passenger targets. Here, I'm also going to show you what the results are when we actually take the triple-negative breast cancer, but this time compared to a normal tissue. The first therapy is cetuximab and gefitinib, which inhibit the EGFR, based on prior data that EGFR is overexpressed in triple-negative breast cancer. Interestingly, when we compare to microdissected normal breast, we don't find EGFR on average to be overexpressed. We actually find no differential expression. This is an observation that was confirmed by qPCR and also by CLIA-based IHC staining. And clinical trials testing EGFR inhibitors in triple-negative have all been negative. The agents imatinib and dasatinib, which target the c-KIT receptor, also based on prior data that c-KIT is overexpressed in triple-negative breast cancer. Compared to normal breast, we actually find it not to be overexpressed, actually significantly downregulated. Interestingly, the normal breast ductal epithelium expresses high amounts of c-KIT, and 2 clinical trials testing these inhibitors in triple-negative breast cancer have been negative. The PARP inhibitors, drugs like nipirib, olaparib, and rucaparib, and many others that are currently in clinical trial that target the PARP. PARP is a DNA repair enzyme, and its repair rationale is based on overexpression of PARP, and also via synthetic lethality, especially in BRCA1 carriers in DNA repair. We find PARP actually to be overexpressed in triple-negative compared to normal, with a 4-fold overexpression with significant p-value. And actually, PARP inhibitors are the only agents to show some positive activity in triple-negative. The results have been slightly mixed based on seeing more activity in BRCA1 carriers versus those who are not, and also some recent work describing potential lack of potency in some PARP inhibitors. But some newer PARP inhibitors that are on the market are beginning to show activity in this disease and are seemingly right now to be a promising candidate for targeting triple-negative breast cancer. So the question is if some of those targeted therapies I showed you before, like EGFR and KIT, are not the target agents we need to use, so what are the right targets? So one of the other analysis that we performed using this data set was to perform pathway analysis, trying to identify novel therapeutic targets for this disease. Here I show you the PI3K pathway, PI3K-AKT pathway in our data set, where those genes in red are those that are overexpressed in the triple-negative breast cancer versus normal. And here we can see many genes involved in PI3K-AKT pathways overexpressed in this disease. To test the sensitivity of triple-negative to PI3K inhibitors, we tested a panel of 7 triple-negative breast cancer cell lines against a panel of variety of PI3K, AKT, and dual PI3K-mTOR inhibitors that are either in phase 2 or phase 3 clinical trial in triple-negative breast cancer, in actually all cancer patients and pan-cancer patients. What we saw very nicely is actually a strong sensitivity of triple-negative breast cancers to these inhibitors. Here is actually the cell line MB231, which was actually the most resistant cell line to these inhibitors. And even in the most resistant cell line, we see nice sensitivity of these cells to these PI3K, AKT, and mTOR small molecules, such that when we test these inhibitors across the triple-negative breast cancer cell lines, we see many of our results in the nanomolar IC50 levels. But as we know with cancer, hitting one target rarely works. Even if we see a response, the tumor does become resistant, engendering the possibility of the promise of doing combination therapy. So we set out to use the same transcriptome sequencing to see if we can discover rational combinations. As I mentioned to you before, also, that genes like EGFR and KIT have failed in clinical trial, and one of their promises is because they're powerful molecules, cell surface receptor tyrosine kinases. So we decided to look at actually the whole genome expression of receptor tyrosine kinases in our data set. Here is a volcano plot plotting the fold change of these receptor tyrosine kinases of triple negative versus normal on the x-axis, and the p-value of the differential expression on the y-axis. As you can see on the top left, genes like KIT that were tested before in clinical trial and EGFR here are either significantly downregulated or not differentially expressed. What's interesting is the genes on the top right of this feed. Here, genes like the receptor tyrosine kinase PTK7 and FRNB2. And actually, prior to this analysis, we were not very familiar with this protein PTK7. What it is a canonical Wnt co-receptor present on the surface of cells and was first discovered to be overexpressed in colon cancer tissue and not normal tissue. And its primary role is involved in epithelial planar development. This led us to delve deeper into the Wnt pathway. Here we show a canonical pathway analysis comparing triple neg normal here on the Wnt pathway, and we find a variety of genes involved in the Wnt pathway to be overexpressed in triple negative breast cancer. This observation led us to test the combination therapy of PI3K and inhibiting PI3K and Wnt. We sought to find a Wnt pathway inhibitor available, publicly available, and we found this Wnt pathway inhibitor, FH535, first developed in 2008 at the Fred Hutchinson Cancer Center, and is a potent inhibitor of the Wnt beta-catenin and PPAR alpha and gamma signaling. What was interesting is when we put these drugs together. Here again, the same resistant cell line MB231 treated both with the PI3K inhibitor BKM120 and the Wnt inhibitor FH535. In purple, you're seeing a log dose increase of BKM120. In blue, you see a log dose increase of the cells treated with FH535. And in the yellow, you see the combination where BKM120 is held at a very small concentration of 10 nanomolar with increasing doses of FH535. What's very interesting is we saw extensive synergy across all 7 cell lines when putting both these inhibitors together. Interestingly, with IC50s approaching single-digit nanomolar levels. So what we're seeing here is a very strong synergy of inhibiting these two powerful pathways together against this disease. To get an idea of why this combination is seemingly so efficacious, we performed an RNA-seq experiment, again going back to transcriptome sequencing, where we took triple-negative breast cancer cells and treated them with the PI3K inhibitor BKM120, and then performed RNA sequencing before and after treatment. What was very interesting was to see that the important genes involved in the Wnt pathway were actually becoming overexpressed or induced, their expression induced by the presence of BKM120. Meaning, when the cells were treated with PI3K, when we knocked down this overexpressed PI3K, the Wnt pathway genes were becoming overexpressed, and such that when we were hitting the Wnt pathway with the FH535, we were knocking down both pathways and potentially inducing a synergistic reaction in cell viability. Very akin to the game of whack-a-mole, where one hits the mole in one with another mole popping up, and then we hit both. Here, genes like porcupine, which is a Wnt maturation protein involved in its palmitylation, being overexpressed 8-fold, and also several cell surface Wnt receptors like Frizzle1, Frizzle2, and also PTK7. This work is now actually being further taken with a combination of inhibitors that target PI3K and also porcupine, now being tested in vivo in patient-derived mouse xenografts. Another aspect that we took advantage of RNA sequencing was to actually study the heterogeneity of this disease. As I mentioned in the introduction, DNA mutational-based heterogeneity is being actively studied in a variety of cancers, and we wanted to see if we can apply the same heterogeneity concept to RNA sequencing in triple-negative breast cancer. So what we did in this analysis was to take all of our 94 triple negs and compare each one individually to the panel of 20 microdissected normals. And what you have plotted here on the x-axis is each of those individual triple-negative breast cancer, and the y-axis the number of differentially expressed genes that are different between each of those individual triple-negatives. And what we see is a wide variety or wide range of the number of genes that differentiate each individual tumor, such that we have roughly at the high end, roughly about 3,600 genes are differentially expressed between a triple negative and a normal tissue, that range all the way down to as little as 1,300 genes. And what we see is actually a wide swatch of heterogeneity in the number of transcriptional dysregulated genes in our data set. We've internally referred to this as transcriptional chaos. What's further interesting was when we correlated this transcriptional chaos, i.e. the number of differentially expressed genes per triple negative, to the total number of non-silent DNA mutations from TCGA. So what we did was to download the non-silent DNA mutations from the Broad Firehose application to the TCGA, and then perform a correlation analysis of transcriptional chaos. And what we saw was a correlation between the burden of the number of non-silent mutations of TCGA translating into the number of differentially expressed genes in transcriptional chaos. What also was further interesting was how this transcriptional chaos actually explains overall global transcriptional heterogeneity. So here you're seeing an unsupervised PCA plot of our 94 triple-negative breast cancers, where on the left you see the triple-negative breast cancers colored by the bottom and top quartiles of the transcriptional chaos, and on the right separated by known triple-negative breast cancer molecular subtypes. If you were to rotate this PCA plot, if we were able to do that during this talk, you would see that the known molecular subtypes, the Vanderbilt TNBC types, do separate the triple-negative breast cancers into their 7 different subtypes. But we also notice is that the transcriptional chaos on its own also separates the triple-negative breast cancer into these 2 groups, just based on the number of differentially expressed genes that separate each individual triple-negative to normal, engendering that in addition to known molecular subtypes, that transcriptional chaos plays an important role in understanding the heterogeneity of this disease. And we have quite a bit of more further data not presented here showing how this transcriptional chaos plays a role in differentiating the heterogeneity of triple-negative breast cancer. As I also as mentioned before, we see, again, a wide range of the number of genes that differentially express and mediate this chaos of 1,300 to 3,500 genes. And interestingly, this represents about 14,000 genes in a genome. As you can imagine, there's 20,000 roughly or so expressed genes in the human genome, protein-coding genes, and this transcriptional chaos analysis encompassed roughly 14,000 of those. We also, as I mentioned, this chaos is dictated by mutational heterogeneity, and definitely dictated by a variety of common mutations like p53, PIK3CA, RB1, and MYC, as I mentioned before, but also many other less frequent mutations. This led us to ask this question that in the midst of this chaos, is there any commonalities that act as denominators of this chaos? What was interesting is when we looked at the analysis, we found actually only 146 genes, what we refer to as the core genes, that were differentially expressed in greater than 90% of the triple-negative breast cancers. Meaning, even though the chaos analysis encompasses as many as 14,000 genes, the denominator between these diseases is very small. Only about 146 genes are analysis. What was further interesting was one of those 146 genes is FOXM1, which we found to be overexpressed 20-fold in these triple-negative breast cancers and activated in our analysis. What was further interesting was when we took this FOXM1 and performed a local network analysis, what we found was when we just focused on those genes, of those 146 genes in that core analysis, we found FOXM1 to directly regulate a subset of those 146 genes. Roughly 15 or so genes that are directly regulated by FOXM1. When we took it to the second level, when we took the network analysis 1 hop, we found a larger number of those same 146 genes being regulated by this FOXM1 network. Interestingly, roughly 46 of those 146 genes being regulated by this one transcription factor. So what's interesting is that even though the denominator, the number of genes that are the same across these 146 in terms of transcriptional chaos, 146 genes across these 94, we find that a lot of that heterogeneity is being dictated by one major transcription factor. We've further done additional analyses and found a couple other regulators of those core genes. So I'd like to end off, the title of my talk is about next generation sequencing and translational oncology research. I want to also talk about a little bit about how we're applying next gen sequencing for correlatives in clinical research studies. One of our current studies is actually applying next gen sequencing to a recently completed randomized phase 2 clinical trial of triple-negative breast cancer, or ER/PR-positive HER2-negative with known BRCA1/2 mutations. This is what is referred to as a post-neoadjuvant trial. Here, patients receive neoadjuvant chemotherapy or chemotherapy prior to surgery. And normally, at the time of surgery, there's two responses. One is a pathologic complete response, meaning the chemotherapy had ablated an entire tumor and there's no tumor at the time of surgery. Or the other response is significant residual disease, meaning disease is present at the time of surgery. We know that patients who gain a pathologic complete response do well after surgery. But we know patients who have residual disease tend to do poorly, in many cases, a large proportion of relapse with metastatic disease. So this trial is innovative in a couple of ways. Current standard therapy for patients with triple-negative breast cancer who have significant residual disease after surgery is actually wait and see or physician choice. There is no standard therapy for this population. So this trial is innovative in that it actually randomizes patients to a therapy, in this case cisplatin for 4 cycles, which is a DNA-damaging chemotherapy, or cisplatin plus rucaparib, which rucaparib is an experimental PARP inhibitor for 4 cycles, followed by maintenance therapy of rucaparib for 24 weeks. This trial enrolled 135 patients, with enrollment completed in early June, with primary endpoints of 1-year and 2-year disease-free survival. From this analysis, we are obtaining a variety of biospecimens for correlative genomic sciences. This includes a biopsy pre-treatment prior to neoadjuvant chemotherapy, a specimen of surgery, and also normal blood and plasma to perform correlative genomic sciences using a variety of sequencing modalities listed here. We are going to be applying whole exome sequencing using the ion proton sequencer to discover germline and somatic mutations, RNA sequencing to profile dysregulated gene expression, and also profiling the plasma DNA to detect circulating tumor DNA as a biomarker of relapse. Our goals from this study is to determine the mutational and expression changes in the tumor that mediate resistance after neoadjuvant chemotherapy, meaning can we find signatures or mutations that make these tumors resistant in the first place, such that these patients develop residual disease after neoadjuvant chemotherapy. To determine gene expression signatures and mutated pathways that predict response to the experimental arm of cisplatin and rucaparib. To test germline variation as biomarkers for treatment response. We know that germline DNA variation, particularly how enzymes metabolize drugs, play a major role in our response to both drug efficacy and also to toxicity. Lastly, we'll be assessing the circulating tumor DNA as an accurate biomarker for relapse. One of the challenges when we sequence from clinical trials is that the samples we get can be very difficult to work with and can be challenging. As one can imagine, many of these clinical trials are multi-site trials present across a variety of states and sometimes internationally executed across various countries. And these samples come in all variety of, I would call it, shapes and sizes that are obtained by the lab, such that sometimes we get very good tumors shown in the upper left with plenty of tumor cellularity. Sometimes we get tumors that are like on the upper right that are intertwined with a variety of stromal cells, or sometimes we get tumors like on the bottom here in center, where comprised mostly of stroma, scar tissue, and with very little tumor, making, and many times almost all samples being formalin fixed paraffin embedded, providing an extra challenge for the study of these samples using next-gen sequencing. So as we've embarked on sequencing this clinical trial, we performed quite an extensive quality control analysis to make sure that we can be successful in sequencing difficult samples. Here we show data where we performed proton paraffin RNA sequencing of the same sample with a 1,000 nanogram input, and also with a very small 50 nanogram input of fragmented of FFP RNA. Here we plot the log2 RPKM, or the digital expression values of these libraries against each other, and we demonstrate a very strong, nice correlation between these two libraries, with an R value approaching 0.92. This was very exciting from our standpoint, is that it shows that low input FFP RNA sequencing is feasible from paraffin tissues at very low inputs. And we've actually replicated this with other paraffin tissues and shown that this is actually doable with samples from a clinical trial. Albeit, sometimes, depending on the age of the study, can affect the results of these sort of sequencing studies. But so far, we've had very good results testing RNA-seq on paraffin tissue from clinical trials. And with that, I'd like to end my talk and thank just my wonderful collaborators who made a lot of this work possible. My collaborators here at the IU School of Medicine for Medical Oncology, Dr. Schneider and George Sledge. who just recently moved to Stanford, Dr. Sue Clare, Dr. Lair, Ivan, nephew, Lou, and Bodve, and also members of my laboratory, Jeff Solzak, Rituja Atali, and Brad Hancock, along with the wonderful folks at the Susan G. Komen for the Cure Tissue Bank, who made this study possible by providing normal breast tissues from a wonderful repository available to the public for breast cancer research. Also, for those of you who are interested in a little bit more about what we do, my laboratory loves to maintain a Facebook page with the research and also some of our more fun photos, and please come see us there at Facebook. And with that, I'd like to send it back to Micheline. I thank you for your attention today.